All right, so we are on chapter 8, section 4, and we're going to talk about tumor necrosis factor receptor signaling. So they abbreviate that TNF for tumor necrosis factor, and then if it's a receptor, we just put the R, right? So this is a really, really cool signaling pathway because you have one single receptor that's going to stimulate two completely opposing pathways inside of the cell. And so if you look at our little diagram here, right? So you have the TNF receptors. So when they bind to their ligand, they're going to trimerize, right? And so that ligand is always going to be TNF alpha. This is also TNF alpha. So when TNF alpha binds, it's going to make those receptors, right, trimerize. And then you're going to have some downstream cellular response. And that downstream cellular response is determined by the relative levels of pro-apoptotic or anti-apoptotic proteins. So here, these are our pro and these are our anti, right? These are pro, these are anti. And so depending on how much of each one is there is going to determine what happens. If you have a high level of the pro-apoptotic proteins, then you're going to stimulate proteolytic cascade that's going to basically like chew up everything inside of the cell, and the cell's going to die. And so the second scenario, right, is that you don't have a whole lot of pro-apoptotic, you have a lot of anti-apoptotic downstream proteins. And so what that's going to happen is that's going to block that normal cascade that would happen. And so the cell would continue to survive, right? So it's just preventing cell death here. So either you're going to promote cell death or you're going to prevent cell death. One of the two when TNF alpha binds to the tumor necrosis factor receptor, right? So how does this kind of work? Well, TNF is a homotrimer, which means you have three of the same thing, right? And so immune cells will secrete this ligand, which is TNF alpha. So it comes from immune cells and travels to the target cell, right? And it induces a conformational change, just like what we've seen in all the other receptors before. We have a change in the tumor necrosis factor receptor in what's called the death domain. Sounds very, I don't know, I guess it's kind of appropriate for Halloween, right? So here are our death domains. Our death domains are in the cytoplasmic portion of the cell, right? And so if you have, trying to go back. All right. So it changes the conformation of the death domains inside the cytoplasmic portion of the receptor, right? And so you normally have something there that basically silences the death domain. And so that's the silencing of death domain protein, or the SOD protein. So the SOD protein basically represses the signal. So this is repression of the death signal, right? So you could stay repressed, or you could have a disassociation. If you disassociate SOD, which is the silencing of death domain, right, then you would go through the process of cell death. So if you disassociate SOD, you would go through cell death. So you have these two kind of alternative pathways that could happen. And it all depends on which proteins are more prevalent. Do you have those anti-apoptotic, like the silencing of death domain proteins, or not? And so the cell has these different things in response to environmental factors. And sometimes that's internal environmental factors, and sometimes that's external environmental factors. It just kind of depends. So when we look at kind of how this works, here's our TNF receptors, right? And so what happens is normally you have these SOD proteins that are like basically hiding the signal, because they're bound to the death domains, and they prevent the death domains from signaling, right? And so TNF receptor signaling is inhibited by our SOD proteins. Now, TNF alpha binds, and you can have an association not with SOD, not with our inhibitor, but with something that is going to initiate formation of a downstream signaling cascade. And so that's the TRADD proteins. And so these are the ones that are going to stimulate cell death, right? These are TNF receptor associated death domains. That's what the TRADD stands for, the triad. So TNF receptor associated death domain proteins bind to the death domain. So that kind of makes sense, right? And so it's going to stimulate this downstream cascade. What is this downstream cascade going to signal to happen? Well, it's really going to be what are called caspases. CASPASES, yes, caspases. And caspases are really pretty cool. You've probably heard of them before, maybe. I'm hoping, maybe a little bit. But they are cysteine, aspartate, proteases. So think about this. If you initiate a signaling cascade that is going to result in proteases becoming active that cleave at cysteine and aspartate residues, you're going to start chewing up basically every single protein inside of your cell. So if you activate these caspases, you're going to get this proteolytic cascade. If you have a proteolytic cascade in the cell, it's going to degrade all of your cellular proteins. If you degrade all your cellular proteins, that's it, kaput, you're done. Cell dies, okay? So I don't want you to memorize all the steps inside of the caspase cascade, right? But you do need to know that it is a cascade, and that in order to make this cascade go, instead of going through phosphorylation, we go through cleavage events. And that kind of makes sense for an enzyme that's there really designed to cleave other proteins, right? Well, what kind of function is it going to have? It's going to have a function to cleave. And so you can have cleavage of these proteins into their basically inhibited form, which is the pro, and then you name it. So pro-caspase 8 is cleaved, it turns into caspase 8. Then caspase 8 cleaves pro-caspase 3. Pro-caspase 3 generates caspase 3, and then that caspase 3 is the one that goes out and chews up everything. So they call it the executioner caspase because it goes in and chews up all the cellular proteins. So you have to know that this isn't so much of a phosphorylation event as it is cleavage events. And so then you get all the molecules degraded and your cell is kaput, right? So kind of the question is, why do we have this one receptor ligand interaction that controls both cell death and cell survival? I mean, isn't that kind of like a waste of resources? Why would we have one receptor ligand, right, causing two competing pathways, right? And the answer is that it's really important to be able to very quickly either kill or rescue a cell. If you have a cell that's proliferating out of control, right? A cancer cell that's no longer doing its function anymore, you want to be able to very quickly kill it. And so having this basically molecular switch, this on-off toggle switch for killing or letting the cell survive, allows for a very quick decision and a very quick cell death, which is really good because you don't want those cells to survive. You don't want them to keep going. So it provides an advantage to the whole organism to prevent damaged or inappropriate cells from surviving, because we want to get rid of those. We want the cell to survive as long as it's doing the right thing, as long as it's performing a function for the organism. But the second that it stops, we need a kill switch so that we can get rid of that cell and it does not turn into a cancer cell, right? Okay, so that was a very short section because you don't have to know all the big details on the tumor necrosis factor receptor signaling. So I don't think I've ever had a video this short before. Yay. Okay, y'all have a great rest of your day.